let's discuss the steps in the action potential in this electrical signal that we're talking about so that we can understand how this action potential or this signal moves along the neuron. So if we move the neuron off to the side here, we can zoom in on a little portion here of the axon. When a signal is not moving along the axon, so when there is no action potential, we call the cell that's at rest polarized. And so here we're indicating a person at rest, but what it's saying is when the neuron is at rest, when no action potential is traveling along it, what we see on the right here is the case. Outside of the axon, we have a positive charge, and inside the axon, we have a negative electrical charge. Polar meaning we have two sides, just like the Earth has a north pole and a south pole. Here we have a neuron with a positive outside and a negative inside. This could seem odd, but this polarized state is the resting state. And so if I'm not sending a signal along this neuron, let's say to my muscles causing movement, my neuron is in this situation. In order to actually send a signal along my neuron, I have to stimulate it by undergoing the process of depolarization. What happens is that the positive charges move into the cell, creating a positive inside to the cell and a negative outside to the cell. What happens, as is being indicated here, is that this signal happens in one place on the axon, and then once the positive ions come into the axon and then outside of the axon becomes negative, then the next portion over on the axon can undergo the same depolarization. And so this is a stepwise process where the depolarization moves down the axon one step at a time until it reaches the end of the axon. Once it reaches the end of the axon and we want to get back to the original configuration of the neuron, we undergo the process of repolarization or resting, going back to rest. And how this happens is the positive ions move back outside of the cell and there is a negative charge remaining inside the cell. Again, it happens in a stepwise process, and this takes a little bit of time, and so our neurons take a moment to recharge and get back to their original polarized state, but it happens fairly quickly so that we can then again send another signal. We want to take another look at this and not just focus on the charges, but what's actually moving in the case of the action potential moving along the axon. And so down on the right here we have a legend showing potassium ions and sodium ions and then negative anions. The potassium and sodium are both positively charged and the anions of course are negatively charged. And so when the action potential is moving down the axon, we have the process of polarization where the inside is negative and the outside is positive, because we have sodium on the outside, which is actually not well indicated in this image, and potassium is more on the inside of the cell, but some of it can leak outside, and we have lots of anions in the middle of the axon. So in the end, we end up with more positive cations on the outside and not very many anions, so the outside is positively charged, and relative to that, the inside has potassium and it has negatively charged anions, so it is more negative than the outside. And so this is the polarized state that we mentioned earlier. In order to undergo depolarization, what happens is we need the inside to become positive. Just as we saw earlier, there's a switching where the inside of the axon becomes positive and the outside becomes negative. What happens in this case is that the sodium that's on the outside of the axon actually moves in to the axon, causing more positive charges inside the axon. The axon becomes more positive, therefore, and the outside becomes more negative. Then, to undergo repolarization, we have a couple of steps. The first, as is shown here, is that potassium will actually leak out of the cell, so the outside of the cell, the outside of the axon, becomes more positively charged. A second step then is that there are sodium-potassium pumps in the axon plasma membrane, which will then pump sodium out of the axon and potassium into the axon to set us up in this original state where there's potassium and anions inside the cell and positive sodium cations outside of the cell. So let's take one more look at the steps of the action potential. But in this case, what we're going to talk about just is the nerve impulse, that changing of charge of the outside of the axon relative to the inside. And so what we're showing here is a cell with its dendrites attached to the cell body and the axon traveling down to one axon terminal. But in the cell, we can see at point A, we have a nerve impulse. This is an area where there's depolarization occurring, and so sodium is coming into the axon. At point B, what we see is that there's a little bit of overflow on the left side where there's some impulse, some action potential, because sodium is coming into the axon at point A. Some of it is moving down to point B, and then causing some of the proteins that allow sodium to move in to actually open up at point B. Those proteins open, and then sodium moves into the axon at point B. And therefore, what happens is we have sodium now coming in that area and depolarization at point B. But then the process repeats, whereas sodium comes into the axon, it moves down to point C, flowing through the cytoplasm, and then proteins open at point C, allowing for sodium to enter the cell at that point. And then this process continues in the same way until the signal actually gets down to the axon terminal. Before we discuss what happens at the axon terminal, though, let's explain the role of myelin in nerve conduction. We talked about the myelination or the myelin sheath earlier. We said in the peripheral nervous system, it's actually formed by the Schwann cells. So here we're showing 4 myelinated portions of the axon. There would be much more than that in an actual neuron. And what we can see is that there's a little space between each Schwann cell where the axon is actually exposed. And those spaces are called the nodes of Ranvier. And what these nodes of Ranvier do in conjunction with the myelination relates back to what we saw earlier about the action potential moving down the axon. Because there's the myelination here, as opposed to the bare open axon that we saw earlier, there can actually only be depolarization at the nodes of Ranvier. And so if we take a look here, on the leftmost portion of the axon, we can see that depolarization is occurring there. But then what happens is, as it states here, the nerve impulse must jump from node to node. There cannot be the depolarization and repolarization occur where the myelin has formed. And so, as sodium comes in at the first node of Ranvier, depolarization occurs, and that opens up protein channels in the next node of Ranvier, allowing for sodium to come in and depolarization to occur. And then it opens up proteins on the next node, etc., etc. And so as you can see here, again, the nerve impulses jumps from node to node all the way down the axon, and that happens much more quickly than we saw earlier, where the nerve impulse moved down just to the next area on the axon, to the next area on the axon, and so on. So now we have the nerve impulse move all the way down the axon to the axon terminal. But then how does the signal move from the axon terminal to the next tissue over, perhaps a muscle cell or a gland or another neuron? So we want to describe the transmission of the signal at the synapse. And so what is the synapse? The synapse is the region of connection between a neuron and then another tissue or another cell. So here we're looking at the axon terminal of neuron A being connected to a dendrite of neuron B, but they are connected actually at the synapse. If we take a closer look, we can see again on the left neuron A at the axon terminal and neuron B on the right with the dendrite. And there's a number of steps and stages in here that we want to take a look at. So let's start with step 1. Step 1 is that a nerve impulse travels along neuron A. And so we were just looking at that with the depolarization and so on. And so the nerve impulse travels along the axon of neuron A. Then step 2 is that that nerve impulse comes to the axon terminal, and as you can see here, there are a number of vesicles with neurotransmitter, a chemical that will transmit the signal from one neuron to the next neuron, in this case, or the next cell. And so step 2 is the nerve impulse actually causes those vesicles to come to the surface and fuse with the membrane of the axon terminal. The neurotransmitter is released into the synaptic cleft, which is the space between the two neurons in this case. Step 3 is that the neurotransmitter diffuses across the synaptic cleft and binds to the receptors on neuron B. Step 4 is that the neuron B membrane then depolarizes, sodium is able to then come into the cell, and the nerve impulse moves towards the cell body. And so if there's enough stimulation from different dendrites being triggered, then the cell body will get enough signal to actually have neuron B then undergo depolarization and send an action potential along its axon. If, let's say, this was a muscle cell, then we would have the signal go across to the muscle cell and trigger then the response of muscular contraction, as we saw in the earlier unit. The last part of step 4 is once the signal has actually been sent along on that second cell, an inactivator breaks down the neurotransmitter. And so we can see that at the top of neuron B's dendrite here, where we have the inactivator, acetylcholinesterase, breaking down the neurotransmitter acetylcholine that was released from neuron A once the nerve impulse continued on neuron B. The next learning outcome is to explain the steps in a reflex arc. So what is a reflex arc? Well, you've heard of a reflex before, and so you can see the person here in the image touching a hot iron, something that is going to be quite painful, and she's looking shocked and will probably pull her hand away from the iron. We know this is a reflex, but a reflex arc is how our nervous system actually causes this change of pulling away to occur. And so if we move the person off to the side here, if we were to take a look at her central nervous system and cross-section through her spine, we might see an image like this, where we see her spine on the right, and then we have the peripheral nerves, the nerves on the outside of her central nervous system that are bringing information into it and then information out away from it. So let's start with the skin and the skin receptors. There are receptors in the skin for heat. And so when she presses up against the hot iron, those skin receptors will actually sense that heat and then send a signal along a sensory neuron. The sensory signal will then run up the sensory neuron, travel up through a nerve, and then into her spine. In her spine, there will actually be a neuron. We call it an interneuron. Inter meaning between, and then neurons meaning neurons. So the interneuron connects a sensory neuron that's bringing sensory information into the central nervous system to then a motor neuron that brings a motor signal out of the central nervous system. In this case, the motor neuron is going to bring an action potential signal down to the ends of those motor neurons, and then there's going to be a signal that will go across the synapse from the motor neuron to the skeletal muscle, causing this individual to flex her muscles and move her hand away from the hot iron. So this is one example of a reflex arc and the steps that take place, but let's take a look at each of the steps using a different example. So if you've ever gone to the doctor before and had them use the little hammer tool here to tap on your patellar ligament, we can talk about what they're actually testing and look at each of the steps here. And so the first step is that the doctor taps on the patellar ligament, and what happens is that there's a stretching of the quadricep muscle. And so when that stretching occurs, it actually triggers a sensory neuron, and then an action potential is sent along that sensory neuron up to the spinal cord. That nerve impulse then triggers a motor neuron, and the motor neuron has an impulse travel along it to actually go back out to the muscle. What happens then is the leg moves up because the muscle contracts. And so what the doctor is actually testing under these circumstances is not how quickly you can kick your leg or anything like that, but it is an unconscious motion. The doctor taps the ligament. There are these stretch receptors that send signals up to your spinal cord, and then motor neurons bring signals out, causing your muscle to contract and your leg to move up. And so this is one way your doctor can actually test to make sure that your nerves are functioning properly and make sure the timing in terms of your reflex here is actually appropriate. So there's two examples of reflexes and looking at the steps of the reflex arc. But let's talk about a couple different types of reflexes. And so we have what's called the withdrawal reflex. And we've looked at that already. The person is touching the hot iron, and then this causes the withdrawal of the hand to move away from the stimuli. The second is the knee-jerk reflex, which we just saw in the previous example. But we also have the pupillary reflex. And so what the image is showing here is when we flash a light into an eye, the pupil will actually constrict, closing down and getting smaller. The Babinski reflex is another type of reflex where you actually use pressure on the bottom of the foot and it causes people to actually contract their foot as a reflex. And so again, we can test, is their response normal and is their nervous system functioning properly?